Hello guys, welcome to our channel. In this video we are talking about gram staining. First of all, gram staining is a method of differentiating bacterial species into two groups, gram positive and gram negative. The gram staining is almost always the first step in the identification of bacteria. It is a valuable diagnostic tool in both clinical and research purpose. Not all the bacteria can be classified by this technique. This gives rise to gram variable and gram indeterminate groups as well. This technique helps us to identify gram positive or gram negative, as well as the gram variable and the gram indeterminate. Now, what is gram positive bacteria and gram negative bacteria? Gram positive bacteria are those bacteria that gives positive results in gram staining procedure. Gram positive bacteria takes up the crystal violet stain in the test as gram staining test, and as a result appear to be the purple color. This is because of thick peptidoglycan, which is the cell wall layer of bacteria that retains the stain after washed away from the sample, like decolorization stage in which we use the alcohol. The examples are staphylococcus and streptococcus. Now, gram negative bacteria. Gram negative bacteria are those bacteria that gives negative result in gram staining procedure. These bacteria are in red color because the organism cannot retain the crystal violet stain after decolorization step. Their peptidoglycan is much thinner as gram positive bacteria. When alcohol as a decolorization that degrades the outer membrane of gram negative, making the cell wall more porous and incapable of retaining the crystal violet stain, as peptidoglycan is much thinner and is present between the outer membrane and the inner membrane of bacteria. So they take up the counter stain, which is the safranin, and appear as red and pink color. The examples are Bacillus and Pseudomonas. Now, this is the image of cell wall of bacteria, gram positive and gram negative bacteria. As you can see in this image, this shows that the gram positive bacteria have thick peptidoglycan, so they can hold or keep the purple color crystal violet, and because of thin peptidoglycan in gram negative bacteria, the organism become more porous when using alcohol, and they appear as red or pink color because of the counter stain, which is safranin. And in gram negative bacteria, the peptidoglycan is sandwiched between the outer membrane and the inner membrane. Now, the reagents of gram staining. These all are used in gram staining procedure. Crystal violet, which is the primary stain, gram iodine, alcohol, which used as the decolorization, and safranin, which is the counter stain, and water. Then steps of gram staining. Firstly make the bacterial smear, which I have already told you in my previous video that how to make the bacterial smear. Then heat fix the slide by passing the slide to the flame for 2 to 3 times. Next is then apply crystal violet stain, which is the primary stain for gram staining procedure, for about 1 minute. Then drain off the excess stain. Then apply gram iodine for about 1 minute. Leave it for 1 minute. Then rinse it with water the flowing water. Then the next step is dip the slide in alcohol for about 2 to 3 times, not longer, for 2 to 3 times, for just a second. Then rinse the slide with water, and the next step is apply safranin, which is the counter stain, for 1 minute. Then wash off the counter stain safranin with water. Then air dry the slide, and when it's dry, then observe in the oil immersion lens microscope by adding the 1 or 2 drops of oil immersion. This is this is the procedure of gram staining, and as you can see, these are the steps of gram staining procedure. After preparing the smear, then fixation step, crystal wallet, iodine treatment, decolorization, counter stain with safranin. As you can see, the left side gram positive